Hello, I'm Bellon. I'm a clinical pharmacology lead at Debiopharm. We try to assess in patients the impact of food and high gastric pH on the bioavailability of our product. As you can see on figure 3, we had 17 patients that were included in our study, and they underwent 3 different cycles. The first one being our control, the second one after having taken food, the third one with a proton pump inhibitor that is increasing the gastric pH. And we can see that the food did not have any effect on the exposure, while the high gastric pH after the proton pump inhibitor reduced by 40% the overall exposure, showing that the PPI are actually to be prohibited around co-administration of Debio 123. Now, with those results, we are able to say that patient can eat at time of Debio 123 administration, which is better for its well-being and actually for the compliance during treatment.
Hello, I'm Noémie Luong. I'm an associate principal scientist at Debiopharm. We work on Debio 432. We want to know how it inhibits USP1, how it works in cells and in tumor models to develop it for cancer patients. So on this poster, we show that Debio 432 is very active on USP1. We also show that it's very selective across a panel of other USP1. So we have tested this model in vivo in a breast cancer model, and we show very good activity, including dose exposure activity. We also show that the pharmacodynamic marker PCNA is modulated in a dose-dependent fashion. We also have a second model where we show that we are also active, and one of the other USP1 inhibitor in comparison is a bit less active. With these good results as monotherapy, we are now preparing the molecule to enter clinical development.
Hi, I'm Luke Piggott, Principal Scientist, Debiopharm. Sacituzumab govitecan is an antibody-drug conjugate conjugated to the SN38 payload, which provides DNA damage to cancer cells. This drug has shown a lot of promise in clinical studies so far, but there still remains an unmet need to improve responses and lead to better overall survival for these patients. The main results were in vitro. We demonstrated strong synergy between our WEE1 inhibitor Debio 0123 and the antibody-drug conjugate sacituzumab govitecan in multiple different models of breast cancer, GBM, colorectal cancer, as well as lung cancer. And we then went on to demonstrate that this synergy is consistent in vivo, where we saw strong enhancement of the effects of this ADC, both in the instance where there was a high expression of the receptor to which it's targeted, as well as models where the receptor is low expressing. This strong preclinical evidence supports the exploration of this combination clinically in patients to see if they would also benefit, as we have seen in our preclinical models. One particular interesting aspect that could be explored is, given that Debio 0123 is a brain-penetrant compound and sacituzumab govitecan has also shown to have effects in the brain metastases of patients, is to see whether or not Debio 0123 can make that response even more pronounced.